Okay, welcome to the first of your video lectures. Should be 3 of these in total, hoping to keep them a bit short, but going over essentially experimental design, stuff you should already know. Hopefully most of this is review, and then a little bit of statistics, might be review, will probably be some new stuff. Let's get in. This one is just going to cover experimental design and controlled experiments. So we're going to talk a lot about various controlled experiments throughout the year. The AP test loves to just describe experiments and then ask you to identify factors about them. So when we talk about a controlled experiment, we just mean some test under controlled conditions, meaning we're holding most of our variables constant and only changing one or two things between trials and tests. So the components of a controlled experiment are your independent variable, which you can think of as the manipulated variable. It's the one that you have control over and that the scientist themselves sets. The dependent variable can also be known as the responding variable. It is what responds to the changes that you make in the independent variable, and this one has to be measured. Then you have your, you have your control groups that can be split into positive and negative control groups. We'll talk about them more in a bit. Your experimental groups, the ones that you actually run the tests on and that you're changing the variables within, the things that you don't know what's going to happen. And then you have all of your constants. These will sometimes be called controls, but do not mix them up with control groups. They are different. These are just variables, things like temperature, time, stuff we often don't even think about as being controlled in an experiment, but things that we are keeping constant. Like if I'm doing a trial of plant growth, I need to make sure that I give them all the same amount of time to grow. That would be a control or a constant. So before the experiment even starts, we need to generate a hypothesis or testable prediction to address some question that we have, and then we will develop the controlled experiment alongside this hypothesis to test whether or not it is correct. Usually there's a bunch of research done before even doing the experiment so that you can actually come up with a good hypothesis, because that can be tricky. It needs to be testable. It needs to be something that you are able to observe. So you usually need to do research beforehand just to show you how you should even go about coming up with a hypothesis. Let's go through an example. So we have goldfish, and it seems like it's behaving strangely when we change the water. We can hypothesize the change might have to do something with the temperature of the water. It's generally colder coming from the tap, and then it heats up when it's in the tank, and that the fish's behavior might be changed to, or might be tied to a change in its respiration rate, its breathing rate. So do some more research, and we find that we can measure the respiration rate basically by counting how often its gill flaps open up each minute, which is essentially the same thing as breaths per minute. So we test the hypothesis, we set up a controlled experiment, we get 5 fish, all the same species, all roughly the same size, so we're controlling for the species type, we're controlling for size, and then we put them in a large bowl together with a water temperature of 20, with 5 other bowls at various temperatures from 10 to 30. The only thing that's different about the 5 bowls is the temperature of water. Otherwise, everything else is the same and everything else is controlled. So during the experiment, they measure the respiration rate of the fish in the large bowl and then immediately put them into one of the small bowls and measure their new respiration rate. And then repeating with all of the fish in all of the bowls, hopefully to measure a change in respiration rate for each fish. So we do all of that and we get this summary. At 20 degrees, that is normal. So at 10 degrees, they take 15 fewer breaths per minute. At 15 degrees, they take 10 fewer breaths per minute. 20 is the same thing as the control, but it is good to note that they still have 0 breaths per minute. This 20-degree testing group would be an example of a control group. not a control variable, but a control group, because you are still wanting to test that maybe it's something about transferring the fish to the new bowl that is causing this change in respiration rate. So if we transfer them to a bowl of the same temperature and don't see any change in respiration, that tells us that we're not affecting the experiment in some way that we aren't considering. And then the respiration rate increases when the temperature increases. So when it gets hot, they breathe faster. When it gets cold, they breathe slower, which is essentially what this says. So if we look at all of the components of that experiment, our independent variable is the water temperature. It is the thing that we had control over. We set up those 5 bowls. We picked the 5 different temperatures that we wanted, and we placed the goldfish in. The dependent variable, the responding variable, the one that we measured, was the change in respiration rate, right? We had to measure it. We had to count it. We had to do tiny bits of math. It depended on the independent variable, not vice versa. So independent variable was the water temperature. Dependent variable was the change in respiration rate. If we think of our constants and controls, all the variables that are identical between the two, we have the fish species, the size of the fish, the size of the tank, the original temperature, the size of the new tanks. We wanted to make sure that the change in respiration rate would not be due to any of those factors, so they were kept identical. Then the experimental groups, again, we can have multiple of them. In this case, we had the 5 different fish that each went into the 5 bowls. And then our control groups. These are groups that do not receive whatever the independent variable is. So in this case, it was a change in temperature. We considered the baseline to be 20 degrees, and so the control group should just be at the baseline. We need some measurement to compare everything to, because we don't know what, well, one, if we're calculating a change in respiration rate, we need to know what we're changing from. But if we just put the fish in hot water and counted its breaths as 30 breaths a minute, we have no idea if that's high, low, or whatever. So our control group is trying to tell us, what does normal look like? So what does a normal fish in normal temperature look like? How fast does it breathe? There can be one control group, there can be many. In this case, we had a positive control group. You could do a negative control group. An example in the negative for a negative control group here would be a fish that gets no water and can't breathe. But obviously that is inhumane, and we know what will happen. The fish will stop breathing. So a positive control is showing you what does normal results look like. A negative control is supposed to show you, like, what does an absence of results look like. Let's talk about them more. Negative control groups also don't get an additional treatment. They are generally identical to the experimental groups. They just don't get the independent variable. If you're thinking of like drug trials and testing new medicines, a negative control group would just be your placebo group. The group that is not getting any treatment, we're just seeing what happens. The positive control groups do get an additional treatment that we know what the effect will be. So they're still not receiving the independent variable. The goldfish still was not in the positive control, was still not given a different temperature. They just get something else that we know what the effect will be, because we're just trying to show that everything in our experiment works. The setup works, the equipment works, we are able to actually measure data that all makes sense for what we're doing. So your positive control group in a drug trial for new medicine would be something like comparing an experimental medicine to some existing medicine that's already on the market. You know that this existing medicine works, and so you're comparing your new medicine to the existing working one and to a placebo. So you're trying to say it's better than nothing, and it's at least as good as what's already out there. Those are the kind of the point of the two groups. So more examples. Control group, again, was the goldfish. You could add another control group where you're just trying to increase the respiration rate of the fish. So you could give them caffeine. I don't really recommend it because it is technically poisonous to a lot of animals, and you could kill the fish. But It should increase the respiration rate, and you could do this and would maybe want to do this so that you could tell if it is possible to even increase the fish's respiration rate. This is all assumed but stuff, so if you put caffeine and increase the respiration rate, you're just proving that, yeah, you can actually see an increase in respiration rate occur. Okay, let's talk about hypothesis. Again, your hypothesis needs to be testable. It needs to include your variables, independent and dependent, and it needs to make a very specific prediction about the outcome. Usually we use if-then statements. You don't have to, but structurally it's if I do this, then this will happen. So if we look at a weak hypothesis would be if the water temperature changes, then the respiration rate of the fish will change. It is testable. We can measure the respiration rate. We do know what the independent-dependent variable are, temperature and respiration rate. The issue is it's not specific. How are we changing the water temperature? How will the respiration rate change? I see this mistake a lot where the question on an FRQ will ask you, like, how did something change in relation to something else? And people will just say, when this changes, the other thing changes. And you need to be specific. What direction is it changing? Is it increasing? Is it decreasing? So a better hypothesis would be if we decrease the temperature of the water, then the respiration rate of the fish will increase. Obviously, with the experiment, we know that was not what happened, but that doesn't matter because we're just trying to test it. So, case study, you should have a link to this both in the worksheet and I link you to the slideshow that has this link in it. It's just going to ask you a couple questions. Read through the case study. It's a quick one page, couple questions, and that is it. And that is your first video. There we go. That's all, folks. First video. Next ones will cover some more of the statistical stuff to help you do the rest of that worksheet. Bye.